through the video. Okay, so I'm going to go back up a little bit in RNA-Seq Analysis, because now that you've done lab, I'm hoping that like a little bit more of a run through this is good and helpful now that you've gone through it once and actually done this once. So the thing that one of the things that we really want to be able to do from RNA-Seq is to quantify gene expression. Now, this isn't the only thing that we want to do from RNA-Seq. This is the main thing I emphasize in lab. That's kind of the main thing I'm emphasizing here, because that is one of the predominant things that people do with this. However, you can also do a lot of other things. Like, you can be looking for alternate splicing variants in genes. You could be looking for RNA editing. You could be looking for all sorts of modifications to mRNAs as well. You can be, I mean, there's so many things that you can be doing with RNA-Seq that are apart from just doing gene expression. But for sake of time, that's where I'm really kind of focusing on right now. So, and to really give good quantification of gene expression, you need to have normalization that's going to happen to that data. So, again, reiterating, longer transcripts are going to get more transcript reads. So the longer a 10,000 base pair transcript, after you fragment it and start sequencing those fragments, there are going to be more fragments to sequence than 100 base pair fragments. So just because of the length of the transcript, we are going to bias how many reads we have for that transcript. Then also just more total reads. Like, for instance, in your data yesterday in lab, one of the sets of the samples, or one of the tissues, only had somewhere in the neighborhood of like 200,000 total reads for each sample, and the other one had about 1.6 million reads. So just that as well is going to bias the number of reads that you have per individual per each transcript. And so what we generally do then is take this and take reads per kilobase per million reads for single-ended, or fragments per kilobase per million reads. The reason for this is because if you do paired end, you're actually getting 2 reads for every fragment that you get. For single read, you're getting 1. So remember, essentially we have our thing here, and then with single read, we're going to read like 100 of these. And with a paired end read, we get a read something, and then a read here. So we have a read this way, we have a read this way. So we have 2 different reads for every fragment that we make. So that's why for single pair or single-ended and paired end sequencing, we do 2 different kind of units for that. But also we have there are ways that then we can calculate that if we have all of these reads, we can calculate, there's different statistical methods, and I didn't have you do this in lab, but you can go on to actually calculate how many transcripts that likely represents. So you can get this to transcripts per million transcripts. And then trimmed mean M value is also reported oftentimes too, which is essentially the same as transcripts per million, but we have some other corrections for biases as well. And so the thing that we had you do, or I had you do, talk about we, myself is we, me and all my personalities. We had you do things. That sounds like the beginning of a really bad horror movie. So anyways, we went through and did DEG, or differential gene expression, or differential expression of genes analysis. And so essentially what we're doing here, is compare the expression levels between samples, and we do some sort of statistical analysis that gives us some ability to detect whether or not this variation between expression between 2 different samples, or a set of 2 different samples, is more or less than you would expect by chance. And so the one that we did yesterday was negative binomial distribution. That is the test that we used yesterday. And then the Poisson distribution test is one that has been previously used that doesn't work quite as well. However, all that being said, you notice what we had was you had a raw p-value, but you also had an adjusted p-value. And there's actually a bunch of different ways that you can run that adjusted p-value. We had it going one way, but you can use a Bonferroni correction. You can use a whole bunch of different corrections that adjust that p-value, and the reason that we have to adjust it is we are making tons upon tons upon tons of comparisons. This is normally really bad in statistics. You don't want to do multiple comparisons, or you want to get this down to as few comparisons as you possibly can. The reason for that is if we're having a error rate of, or essentially we're setting our alpha to 0.05, or a 5% chance of something happening, or seeing some distribution by chance purely alone. That's essentially a 5% chance. Now, if we do that over the millions or hundreds of thousands of genes that we're looking at, you will get false positives like all the time. 5% of 500,000 is like 20,000. That's a lot of genes to have like just false positives. So one of the really big problems with just doing this kind of thing, and one of the really kind of major hurdles in any DEG analysis is controlling for a false discovery rate. And so this is a really big problem that we actually didn't go too much into lab. I had you guys run, we do have one level of buffer against false discovery rate, and that is the adjusted p-value. Oftentimes, if you were going to go publish this data, that is not sufficient. You would have to do some additional work to protect against that. One thing that people do, actually, is start out using RNA-Seq. So they actually go and essentially we can screen for almost every gene and see what looks like it's differentially expressed. And this will narrow it down. And then once you have like a handful of candidates that are looking good, then follow that up with qPCR. qPCR, if you're not familiar with it, being essentially PCR in which you can quantify how much of the original template that you had. So it's another way of quantifying how much DNA was in something of some particular sequence. The difference is that we are targeting specific transcripts. It actually quantifies a lot better than RNA-Seq does. We can quantify a lot more accurately with qPCR than we can with RNA-Seq. But the difference is that you can't do everything. You are targeting one specific sequence. So what you can do is, okay, so we find a handful of different things that we think are very likely upregulated or downregulated in a particular tissue. We can go back then with qPCR, verify that, and if your RNA-Seq and your qPCR are giving you the same answer, then you can be fairly confident that this is a real up or down regulation. So, but keep that in mind, is that if you see a study in the future that's just running RNA-Seq with very little false discovery rate control, they are probably finding lots of things that are not like just random statistical noise in the data, because you run this many comparisons. You are finding hundreds of thousands to sometimes millions of different transcripts. And by transcripts, I'm not saying like the individual reads, I'm talking about the actual number of different transcripts. You only have a few 100,000 genes, but after you account for editing and differential splicing and other things like this, you have millions of different transcripts you can possibly have. That is you will find things just by chance. Anyways, we have ranted enough about that now. But that's one thing that just in general. Also, replication is really important as well. So you can't just really compare one sample to one sample. Now, we did a really light thing in lab where we had 2 samples versus 2 samples. Usually it's recommended that you have at least 3 samples for each different tissue or organism or whatever you're doing. For each group that you're doing, to at least do 3 runs of that. That seems to be kind of like the lower threshold of what you should do. Okay, so DEG analysis is not the only thing that gets done. We can also do discovery of different isoforms. And this can be integrated with differential expression studies, and to really see what is the differential expression of these different isoforms. So you can both discover what the different isoforms are, and then also look at this. Now this relies on a little bit different statistical approach, which I'm just going to, like, in principle, tell you about. So imagine that we have a gene that has exon A, intronic region, exon B, or exon B, intronic region, and C. And imagine this comes out, there's two variants in reality. We don't know this, but there's two variants. You can have transcript AB, or we can have transcript, let's say AC. So these are the two different ways that this can be alternately spliced in reality. What's going to happen is if you do really low coverage, then you're going to get some reads here, you're going to get some reads here, read 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 here. Same thing with this. So we're going to get all these different reads from different fragments here. The thing that you really need for this is there's only a certain number of these transcripts that are actually useful for informing you about the different levels of these isoforms. And those are the ones that cover the intronic region that was cut out. So right here, probably this one, this one, this one. And this is imagining we do not know what isoforms are present. So we don't know if there is also BC or ABC. We don't know if those are present. This is what's actually present in reality, but we don't know that. So the only ones that are really informative for that are these coverage ones. Now, well, directly informative for that. So you need these ones that cover these junctions between exons. And so you can quantify from that. The other thing that is really helpful as well is imagine this one is expressed 10 times more greatly than this one. You can also look at your relative transcripts. If you look at the number, you have really high amount for A, it'd be slightly lower for B, and then not very much for C. And you can get a decent idea for that. So oftentimes when we're doing this, we take these two different lines of evidence, the relative proportion of each of the exons, and then also information about individual reads that span these exon junctions. And build a model of how many, well, first off, which different isoforms do we have, and then also the relative proportion of those. I'm not going to go too deep into that, just kind of give you the kind of sense of, in general, how this is done. So there's a couple different things that we're doing to kind of improve or do RNA-Seq better. You know, one of these is single-cell RNA-Seq, which is relatively very new and very interesting. But this allows us to get the transcriptome of a single cell, which is nice. And we've used this to, first off, oftentimes we look at certain cell types and just from morphology or apparent function or some other things like this, expression of a few proteins, we consider some certain cell types to be fairly homogeneous. It actually turns out that we are now finding that they are not, that there's lots of very kind of cryptically specialized cells within seemingly homogeneous tissue, which is interesting. Also, that even in what you expect to be like the same tissue and fairly homogeneous cells, that we're having somewhat randomness in gene expression between otherwise identical cells, which is really interesting as well. And then, but the point of this is that in isolation, a single cell RNA-Seq is not particularly helpful. It's mostly useful only when you're comparing this within a population. So putting it into the context of a population of cells. The other interesting thing here is, so we talked about when we were going through sequencing, I briefly mentioned smart sequencing. That's being done by Pacific Biosciences, or what's also known as single molecule real-time sequencing. The really cool thing about this is that their individual reads can be up to 4,000 base pairs long. So when you guys were assembling things, we're dealing with base pairs that are like 50 to 70, maybe more than 100 base pairs long, and then you have to spend all this computational time to assemble those. And then we have to do a lot more. If we're interested in isoforms, we have to do this kind of jumping through hoops to start to determine, based on all of these like small individual reads, which isoforms are present and what quantities. If you can read the entire transcript, this opens up a whole new kind of world of being able to do transcriptomics, much computationally less intensive. So the nice thing here is we can sequence an entire transcript rather than just a single little fragment of it and then keep doing that over and over. Now, the reciprocal of this, or the downside of this, is that at this moment, smart sequencing actually has a fairly low accuracy, which makes it weird that you call it smart. But it seems like smart would be more accurate, but maybe shorter. I don't know. That's just what comes into my mind. And actually, for every individual read, or every individual base call, is something like 87% accurate. So like if you had a stretch of 100 base pairs, only 87% of them would actually be correct, which is really bad for sequencing. This is not that great. But, so you do need to do multiple reads just to get a single accurate read. But the other point here then is you actually, if you have a reference genome, you don't really need it to be all that accurate. You have an accurate genome. So this eases, I mean, it makes it easier to do alignment and reconstruction, and the computation is significantly reduced, and you can have much higher confidence about what transcripts you're finding and where this goes, and how many of something are there, what isoforms are there. However, that being said, because of this low accuracy, at this moment, you can't really do like de novo assembly from this. We're not going to be able to just from scratch assemble a new transcriptome. We really need to have a reference genome that, hey, we have a mostly accurate transcript here. We can map that onto the genome and figure out where this goes, and then get the completely accurate, with a more accurate sequence from the genome itself. But just using these transcripts as like the true sequence itself is not possible at this point. So that's cool. Okay, so when I started up this quarter a long time ago, one of the things I was talking about was that there was being produced like insane amounts of biological information all the time. That we are producing information faster than we can really fully analyze it. And I think this meets its full fruition, like just the claim that this is happening in transcriptomic data. Because it's crazy just how much transcriptomics is being added all the time. There's just crazy amounts of it. And when people analyze, they produce some sort of transcriptomic data set, and they analyze it one way in which they're interested in. And then they go on, and that's all, they put it up online, and then it's deposited in some sort of repository, and then it's sitting there with insane numbers of different analyses that can happen with it that have never happened with it. I mentioned at some point that I thought it's just like a matter of time until some kid living in his mom's basement makes some huge discovery in biology. I think if they do, they're going to be using like freely available transcriptomic data. I'm kind of excited to see that happen because I just want to be right about things, but also I just think that'd be kind of cool. I kind of wish like I could have been that kid in the basement, but there's no transcriptomic data when I was, completely in high school. There was transcriptomic data, but just not like it is today. Which is no, we're just barely getting the human genome project stuff launched. Wow. I normally don't feel old, but sometimes I do. So anyways, there's just tons of it out there, and so what we've done so far in lab and in talking, we've kind of gone from, okay, so we have this sequencing data, and then what do we do with it to the point of getting differential gene expression? But then people do their initial analyses, they deposit this up onto some repository, And then that's available, and there's still lots and lots of science to be done there. There's still lots of stuff that people still do and start their studies with actually looking at what transcriptomes have been produced so far. So there's a lot of publicly available transcriptomics out there, and just like genetic sequence, most journals, when you publish, require you to deposit the transcriptomic data that this is based on some public repository, so it's publicly available, or other people can go back and analyze your data as well. Some of the more common ones that you see is a gene expression omnibus, which is an NCBI tool. It's like GenBank or BLAST, which are all these different NCBI tools, which is run by the National Institutes of Health. There's also Array Express, which is EMBL, the European kind of equivalent of the NCBI. And both of these have just gobs and gobs and gobs of transcriptomic data, and it's most of it is microarray data. However, all that being said, by this point, by the point of having the differential gene expression, the analysis of differential gene expression is going to be relatively similar whether you're using microarrays, RNA-Seq, ESTs, wherever you're using. Not exactly the same, because again, RNA-Seq is giving you more absolute quantification of the transcriptome, whereas microarrays is still giving you relative amounts of that. There is still considerations to be made there about the relative expression or the differential expression, but that being said, it's still relatively similar at that point. So let's take a look at one of these. So first off, Tyler and Nancy, you guys are interested in something for your research. Does either one of you want to just briefly explain what your interest might be in 9L cancer?
Which part? The expression of vimentin?
Yeah, let's talk about that.
Okay. About how the expression of vimentin may be changed in the cancer tumor.
Yeah, so why do you think anything about that?
Why do I think what?
That
Well, the fact that it could be upregulated on the surface because we're finding it on the surface of cells and it's a cytoskeleton protein.
Okay. That's interesting. I mean, so how do you know it's upregulated?
Looking for?
No, this is good. Go for it, Tyler. I don't know, just give us some information here.
It's been observed to be upregulated in carcinomas
Okay.
as a part of the cancer and the tumor metastasizing into other tissues.
Okay.
So comparison hopefully between the two different types of cancers.
Yeah. And then why do you guys, okay, so what brought your specific interest to Vimentin and 9L? I mean, or your labs, I guess I should say, since it wasn't you specifically.
The fact that we think an antibody is binding to it.
Okay. So you guys found an antibody that's binding to something, right, on the cell surface that was specific to 9L, right?
Yes.
And then someone ripped that out and sent it off to be sequenced, and then they found the protein sequence and it turned out to be Vimentin, right? And do you think there's also like a protein or a carbohydrate on there, an antigen, probably? Okay. So that's kind of the basic story. So, okay, so 9L for you guys that otherwise might not be interested. Okay, so this is like a rat brain tumor, and they are really interested in freeing the world's rats of brain tumors because they're really horrible, and we really care a lot about rats. Actually, no, not quite. But 9L is this rat brain tumor. It's a gliosarcoma. So it's predominantly acting on not the neurons, but the support cells in the brain, and then they grow big and bust your brain up, and that sucks. However, all that being said, one of the really interesting things is to try to find these tumor-associated, tumor-specific antigens, so you can kind of identify tumor cells apart from all the other cells, and then you can start to target them and do all these other cool things. So Red's lab has been working on this, and they have found a ways back, Ben McCluskey, but even before that, because I think they found it and then they lost it and then they found it again. Kind of an interesting thing because the freezer broke or something. They destroyed all their antibodies and everything. And that freezers going down is the horriblest thing ever in science. Hey, Jesse Katie's birthday. What do you know? So anyhow, all of that's too bad. But they eventually found it again, and now they have this tumor, at least associated antigen on 9L, and they're trying to characterize it, and they're interested in it here. Do you know of anyone that's published anything about Vimentin being upregulated in 9L?
No.
No. Okay.
Not in 9L.
Not in 9L. Okay. But here's the thing, there's tons of transcriptomics data out there, and even if anyone hasn't published directly on that, there might be information about that out there anyway. So, let's take a look. So, here's what we're actually looking at GEO. This is the Gene Expression Omnibus. Again, this is a tool of NCBI. And it allows you just to go looking around at transcriptomic data that's out there. So, if you come here, under tools right here, and you can just get here by just like Google searching NCBI and Geo, and it pops up. It's like the first thing that pops up on my browser because now that actually Google's been doing like searches like that for a while. Anyways, okay, the very first thing under tools is search for studies at Geo datasets. Hit OK there. So let's just search for 9L. Let's see if there's been any transcriptomic data done for 9L cancer. So we just hit enter. Yeah, it looks around for a while. Lung tissue from smokers with severe emphysema. No. Like, so 9L are just two characters. But hey, let's just go with it. Let's see. Flower development, probably not. Brought. Well, what about this? Rat brain orthotopic tumors. Brain tumors kind of sounds like it's promising, right? Okay, so what we have here is, I don't know if you can read this, it says transcriptomic analysis of early response of rat brain orthotopic tumors after synchrotron. This I gotta look it up here. Synchrotron microbeam irradiation. So, cool. Hey, we have some transcriptomic data here. And it gives you the contributors, so the authors and the citation where this came from. So we have, this is actually PLOS 1 is where this was published. So this comes from a study in PLOS 1. Let's take a look at that. I'm going to go look at this up real fast. I would, English professor, I would be so bad at that. So I'm glad I'm not. I do those things. Okay, so here. So really quickly going through the abstract, it kind of looks like what they did is they took normal rat brain cells and tumor cells, and they irradiate them, and then they look at the transcriptome of non-irradiated ones and irradiated ones, and see how does the response to radiation change between normal rat brain cells and tumor rat brain cells. Cool, huh? Okay. I'm curious if they mention anything about Vimentin in here, so let's look. Vimentin. Nope. Did I spell that right? Vimentin. Nope. Okay, so we go down and look through here, look at the graph. Other stuff here. Here's an expression thing. That it's rats, so I can say it that way. Okay, so this is differential expression data of different groups of genes. So here's the number of genes in the brain. Okay, but it's in large groups. It's not giving us anything. Okay, but we don't really care about what the study said because we're interested in their data and reanalyzing their data. So let's go back here, and then here's the cool thing: is if you go down, you can. There's this thing that says analyze with GO2R. So this is a really cool tool in here that actually, there's several R packages that allow you to directly interact with geo databases called GeoQuery and Lemma. And Lemma is just one that allows you to do the back-end analysis of like the statistical analysis of comparing. Actually, if you get this, and that's specific for microarray data. So actually, if you go up, I looked at this but I didn't show you, it tells you what kind of data this is. So up here it has experiment type. It has expression profiling by array. So this is a DNA microarray, which, hey, we have lots of rat microarrays. That's kind of nice. So we can go down and we can click on and go. What this does is this is actually a like a graphical interface for their R backend, so you can actually do the R analysis by just interacting with the website. And the really cool thing is you can't see up here very well. Okay, so this is. Can you kind of? Okay, so this gives you the accession number. This is every sample that they ran. So this is all the different samples, the title of it, the source, the tissue, the cell type. You can't see it right now, but this says 9L rat cells here, glioblastoma. These are normal brain. So all of these right here that it looks a little bit longer is 9L, and all of these are normal rat brain cells. So hey, we can compare normal rat brain cells. The other thing that if you look over here, these ones have no radiation. These ones have the MRT radiation, no radiation, MRT. So we have essentially 4 groups. 9L that both got the radiation, didn't get the radiation, and then we have normal rat brain cells that didn't get the radiation, did get the radiation. Hey, they don't talk about Vimentin in their paper, but we could go looking for Vimentin now, and we could go looking for it. We don't want to look at the irradiated ones because they're all messed up anyways. Who cares about them? But we can look at the non-irradiated from both the normal and that. So here's what we can do: is up here you can type in. I really don't like the watch it outedness of this. Let's come back here. Is that worse? Yes, that is worse. That is so much worse. That's gone. Okay, never mind. I have. We're so of a creek. Okay, if you really, I'm recording this so you can go back online and look. Okay, so this allows us to define groups. So essentially what we can do is we can do a reanalysis, and we can chunk these into the groups that we want them into. So all I do here is enter in a group name. So I want 9L, hit enter, and it brings up a nice little, can I just highlight this? So it's this little pop-down window. I've made a group of 9L, and now I can just type in another name here for like normal rat brain cells. So I'm going to call them NBRCs. And then all I have to do over here is like select the ones I'm interested in. So right there, I've selected all the 9L rat cells. I hope I am telling you well because you can't see what I'm doing. I just selected all the no irradiation 9L rat cells. I'm going to add them to the 9L group. And it's now highlighted them all a lovely blue color that you cannot see. Very, very pleasing. And now I'm going to select all the normal rat brain cells with no irradiation and add them to the normal rat brain cells. Okay. So now I have these 2 groups. I can go through and look at the differential expression between these because all the transcriptomic data is there for me to mess with. Okay, so then we go down. And there's 2 different things. First off, we want to look at the value distribution, which gives us whether or not there is comparable reads for all of these. You want the distribution of reads to be somewhat similar. And it's going to give you box plots. We hit view. Did I hit view? Yep. Okay, and it says the job submitted. It's going to take a couple seconds. Okay. And it's so washed out you really can't see it. But essentially what you have here is you have box plots. And yeah, they're all looking about the same. So here's the medians of all the box plots. You see it's all about the same. The upper end is up here, and then the boxes all look about the same. I'm sorry that's so washed out. It's hard to see. But yeah, so they're all comparable in terms of the level of analysis or the level of reads that we got. So what we can do here is we can go back to the GO2R. And there's actually, okay, so there's 5 different tabs up here. There's GO to R, value distribution, options, so we can mess with that. We can also look at profile graph, and then the last one is the R script. It will give you the R script that it used to generate everything that's generating. This is actually one of the things with online just kind of GUI interfaces, is that if you use that to produce some data, it's really hard to make that reproducible because all of your clicks and everything are really hard to document and then communicate to someone. With this, one nice thing about Geo to R is it generates the R script that you got everything. So you can just copy that R script, bank that away, and if someone else wants to reproduce what you do, you just provide them with the R script that you generated and they can produce that. So kind of nice. Okay, so what we go is we go here to Geo to R, and it gives us this option of exporting all of the results, so all of the differential expression, and it's going to be similar values to what we were looking at yesterday, or we can just look at the top 250. So let's look at the top 250 at first. We can always go back and look at the total results. Look at that. As processing, this says processing right here with a little ee. It's not really making that sound in case you were curious what it sounds like. Processing. Okay, and what we have here now is a list. It has the gene ID, a p-value, and an adjusted p-value, just like what we were seeing in lab yesterday. And then we have a t and a b value. I'm not going to have you worry about those right now. But then we have the log FC. This is the log base 2 fold change. So we have these similar things that what we were talking about in lab yesterday. They're really important. Adjusted p-value, a p-value, and then log fold change. And then also here we have an ID number. But the more interesting thing here is over here we have a gene title. We actually have like the human interesting name to it. So let's go look in here for Vimentin. Yeah, I saw it pop up before I started. Vimentin. There we go. Pops up right here. Okay, so we have Vimentin there. So there's a little drop down here that we can drop that down. And it gives you a couple different things. So first off, it gives you actually the expression values for this is 9L here. This is normal rat brain cells here. And then what we got up above here is we have a p-value, adjusted p-value of 1.1 times 10 to the negative 8, so very, very different. And we have a log fold change of the normal rat brain cells compared to the 9L of negative 3.33. So 2 to that power is what the expression, the average expression level difference in normal rat brain cells compared to 9L. So, this is something that I imagine you guys gonna do. And the question that you kind of had, and the cool thing is the data for that kind of thing is just out there. So here's some transcriptomics of that. But this is the kind of thing that you can do now that's relatively easy because people are just like racking up transcriptomics data, just like throwing it out like candy. Actually, I wish people did actually throw candy around. That would make life very sweeter. Anyway, maybe I should just do that. Just come to class. Transcriptomic data. Have all the transcriptomic data. Whoops. Didn't hit anyone. I'm good. I mean, right now people are going through biopsies of like tons and tons of tumor biopsies that we have done, and actually like transcriptoming, nicking, like all sorts of stuff. So actually, there's really robust data sets of generally again microarray stuff for lots of different cancers in humans, not in rats. I mean, also in rats. So this kind of data is really prolific, and you have ready access to it. So this is something I'm actually going to go into a little bit more next week, how we actually take advantage of a lot of this online repository database data, and that's what lab next week will be on. So at this point with transcriptomics, we've gone from you have the raw sequence data, like hot off the Illumina sequencer, and we have gone from there to actually getting differential expression. Now what I want to do is then go from that differential expression that people are producing and putting up and being able to then begin, okay, hey, you don't have the money to actually go out and do the RNA-Seq maybe, but you can start mining some of this data that people, like again, Whoa, hey, I got another thing here. Thank you, calendar. Like in this case, where they didn't care about Vimentin, I bet you actually if you go back and you look at the difference in Vimentin, because what they were interested in is like, okay, 9L, after we irradiate them, did Vimentin change? Or the normal rat brain cells, after they are irradiated, the Vimentin change? And what are the differences between those changes between 9L and normal rat brain cells? So if Vimentin didn't change between irradiation and not irradiation, just in 9L, they didn't care. They weren't looking at it this way. This is something that wasn't important to them. It is important to other people. And this isn't something that's going to show up in their paper. It only shows up if you go back to that transcriptomic data that they produce and start looking at that. So, are there any questions with that? Okay. Well, with that, have fun Monday not being here at 8 a.m. And I will see you on Wednesday. And I will probably get up a couple assessments this weekend. And a lot of grading data. Woo! How's it going? Sure. So, the one I wanted here was this one that we made that was wide before you scaled it. So if you go back into your code, like right after you do the, and you can just go back to that code. Do you have like your script up somewhere?
I can be here, I just don't want to. Is that too late? You guys do that? Like, no.
You just run this right here again, and then just hit Y so it actually pulls up on here, and then just go ahead and copy that.
Okay.
Yes.
Did
Yes.
Did you
I
get-
got you. I got your